Celibrizi. Good afternoon to everybody. Today you have the pleasure to receive Professor Agostino Celibrizi from the King's College, London, who will give us a talk named Targeted Molecular Probes in Drug Discovery and Bioimaging. So a little bit about Professor Celibrizi. He has a vast professional experience in medicinal imaging chemistry, and currently he is a senior lecturer in the Institute of Pharmaceutical Science, King's College, London. Professor Celibrizi is a research fellow in synthetic chemical biology, proxomics project. The main interests of Professor Celibrizi is medicinal imaging chemistry. We received many awards, and the last one was from the Center for Therapeutic Innovation, co-lead Medicines Development Unit from the University of Bath in 2022. The professional activities of Professor Celibrizi, among them, he is guest editor for the special issue Serine Protease Inhibitors and Therapeutic Applications in 2022. And the Journal of Chemical Biology, he is an editorial board member in the period of 2016-2017. He has a financial support from Renapharm AB, industrial grant of about £80,000 for the year 2023-2024. The teaching activities of Professor Celibrizi involves Master of Students, Master of Science, Master of Science students and PhD students. And he is a member of the PhD committee thesis. Among the selected conference and seminars of Professor Celibrizi, he is noteworthy to say the 8th International E-Conference on Medicinal Chemistry, held in 2022. About the publications of Professor Celibrizi, he has more than 75 papers and 4 book chapters. In the year 2023 and 2024, he published 3 papers, and one of them was with one of our students, Renan Rodrigues de Oliveira Silva, which was with Professor Celibrizi in the year 2022. So it's a pleasure to have you with us, Professor Celibrizi, and we hope that in the future we could be personally to know us and our School of Pharmaceutical Sciences.
Thanks very much, Mauri. I can start?
Yes, please.
So thank you very much for the introduction, Mauri, and thank you for the opportunity of arranging this seminar online. So in this talk, I'm going to introduce an overview of the research activities that we perform in our group at King's College London within the Institute of Pharmaceutical Science. As a background, we are a group of medicinal chemists, so we develop new molecules and we perform biophysical screening assays to test the activity of these new molecules as therapeutic or diagnostic agents. In terms of research interests, our work focuses mainly in these three areas here. So synthesis, metal chelation, and development of optical probes. So we have developed molecules for a number of biological targets, for example, G protein couplet receptors and other membrane membrane proteins, molecules for that target enzymes, and molecules that accumulate specifically in some of the organelles within the cells. When we identify a hit compound, we translate this hit compound into targeted probes equipped with fluorophores for optical imaging, or introducing into the hit compound metal complexes and metal chelators for imaging via positron emission tomography, so PET imaging. In this talk, I'm going to introduce three projects which are currently ongoing in the group. And the first one will be in the area of G protein coupled receptor research. So we'll focus on targeting a subclass of GPCRs, the formyl peptide receptors, which have involvement in inflammatory conditions. The second project will be on imaging probes for alpha V beta 6 integrin in the context of pancreatic cancer. And the third project will be on oligomeric complexes between iron and fatty acids for the treatment of anemia. So we can start with the first project on GPCRs. Here are just some numbers. So GPCRs are the largest class of membrane receptors in humans, with more than 800 subtypes discovered to date. It is estimated that 30 to 35% of currently marketed drugs have a mechanism of action that influences or modulate the GPCRs, both as primary mechanisms or secondary effects. And also, more than 160 GPCRs are already validated drug targets. But at the same time, it's generally agreed that additional 350 GPCRs are expected to be druggable, although for these many roles and mechanisms are still unclear, and so needs more research, more investigation. So in this context, there is potential for drug discovery in general, and there is potential for the development of imaging probes, because with probes that can help to visualize the receptor, we can understand better signaling and mechanisms of these newer classes of G protein coupled receptors. Our work in GPCR started with a scaffold hopping analysis assisted by molecular modeling techniques, for example, inverse virtual screening. So in this study, we have screened a large library of small molecules ligands based on different heterocycles, as for example, these compounds here in the slide. So this library was tested against a panel of G protein coupled receptors. And we found that molecules based on the pyridoxine, so this heterocycle here, have good affinity In this way, we identified ligands for formyl peptide receptors, as these two examples in the slides, which I will discuss in more detail in this first part of the talk. But we found also ligands selective for other GPCRs, for example, the alpha-2 adrenergic receptor or the adenosine receptors, when the pyridoxine is included in bicyclic or tricyclic structures. With regard to formyl peptide receptors, these are receptors mainly expressed in white blood cells. So, for example, neutrophils, monocytes, macrophages, and other immune system cells. These receptors have key roles in the activation and functioning of immune system cells and are considered involved in inflammatory conditions and host defense mechanisms. There are 3 subtypes in humans of formyl peptide receptors, so FPR1, 2, and 3. And here is reported a proposed signaling pathway to show how these receptors can modulate the actions, the functioning of immune system cells. So, for example, they can promote phagocytosis, chemotaxis, superoxide production, so all actions that are relevant in inflammatory conditions. Most of this signaling pathway needs to be fully confirmed. But what is clear is that an activation of formyl peptide receptors produce activation of immune system cells, and this generates a series of effects in the cells that are linked to the resolution of inflammation. To synthesize pyridazinone molecules to target formyl peptide receptors, we have set up some synthetic methodologies. So here we have, starting from the diterbutyl malonate, we can perform sequentially an alkylation on the acidic carbon and an acylation to obtain this desubstituted intermediate, which is then hydrolyzed in acidic conditions and decarboxylate on these terbutyl esters. And then a sequential hydrolysis in basic conditions of the esters gives the gamma keto acid. Gamma keto acids can also be synthesized from succinic anhydrides via reaction with a Grignard reagent. And from the gamma keto acids, we can perform a ring closure with hydrazine to obtain the pyridazinone, which can then be functionalized to obtain ligands for FPRs. In another synthetic method, we start from beta-diketones compounds, which are reacted with chloroxime in a dipolar cycle addition reaction to obtain this isoxazole intermediate here, which can also be ring closed with hydrazine to a bicyclic compound, and then the ring of the isoxazole is opened in reducing environment using palladium on carbon to obtain this other scaffold as a pyridazinone scaffold. From the scaffold, we have synthesized a library of ligands for FPRs, which have this general structure here. So from the top scaffold in the slide, we can perform a condensation reaction in position 4 with a carboxybenzaldehyde, and then an alkylation on the nitrogen in position 2 to obtain this type of ligands for FPRs. Whereas on the other scaffold, we can first perform an alkylation on the nitrogen in the ring in the heterocycle, and then the amino group in position 4 can be reacted via cross-coupling reaction with a boronic acid in the presence of copper acetate. This gives this other class of ligands for FPRs. We have developed a library of ligands and tested the activity for formyl peptide receptors. Here are reported some example of ligands that we have identified to show that, for example, the ligands in this panel are selective towards only one isoform of the FPR. So they bind only FPR1 or FPR2, and the activity is in the low micromolar range. Whereas in this other panel, we have potent ligands with an activity in the nanomolar range that binds to the 3 isoforms, so FPR1, FPR2, and FPR3. Also, these potent agonists of FPRs resulted also potent in activating human neutrophils at nanomolar level. How we test the activity of the molecules for formyl peptide receptors? We perform functional tests, which is the basic in G protein coupled receptor research. For example, we perform the chemotaxis assay by using chemotactic chambers where in the top compartment of this chamber, we load a suspension of the cells that we want to test. And in the bottom compartment, we load a solution with ligand, so with the compound that we think is a chemoattractant. The 2 compartments of this chamber are separated by a semi-permeable membrane, and so we evaluate the amount of cells that migrate in the bottom compartment following a chemotactic stimulus. In this way, we obtain a value of potency for the agent as chemotactic agent. The other test that we perform is the calcium flux, and this is done in transfected cells or human neutrophils. This gives a value of potency of the agents as an agent that can stimulate the increase of calcium inside the cells. The calcium flux is a fluorescent-based assay and uses a chelator for, we use a chelator for calcium in this test. So this compound, Fura-2AM, in the ester form, is uptaken by the cells, and once inside the cells, the esters are hydrolyzed to a free carboxylic acid form. The free carboxylic acid is trapped inside the cells and can chelate calcium. And both species, so the free acid and the complex with calcium of Fura, are both optically active, but the optical profile is different. So we have an increase of fluorescence for the complex, so for the Fura bound to calcium, and we have a shift of the absorption wavelength for the calcium bound to Fura, which absorbs at 340 nanometers, and the calcium-free Fura, which absorbs at 380 nanometers. So these differences can be exploited to calculate the concentration of calcium inside the cells. This is a typical plot that we obtain in our experiments, where we can see that at the beginning we have the cells loaded with Fura only. And then when we add the formyl peptide receptor ligand, we record an increase of the fluorescence if the ligand is an agonist and so stimulates increase of calcium inside the cells. And from this part of the plot, through a titration experiment, we can calculate then values of EC50 and IC50 for the potency of the ligands in activating calcium flux, so in increasing the calcium inside the cells. Once we have established that this structural motif is a valid binder for formyl peptide receptors, we have used this as an address system to develop optical probes. So in position 6 of the pyridazinone, we have introduced fluorophores for optical imaging. And we have decided to modify the position 6 because both from the modeling and from the screening of the library, we found that this position is tolerant to modifications. And so we should be able not to disrupt too much the activity by modifying the molecules. So we have tested these first two optical probes for activity towards formyl peptide receptors, and the result is that the compound E is highly active with potency on formyl peptide receptor 1 of about 10 nanomolar. So it binds preferentially formyl peptide receptor 1, since on formyl peptide receptor 2 the potency is at micromolar level. Differently, the compound B1 resulted not active as an agonist on FPRs. We believe that this difference in activity is due to the longer linker present in the optical probe E. So the longer linker keeps the fluorophore far from the targeting module, from the address system, and so there is less interference of interaction with the receptors by the address system. In this other molecule, the linker is shorter, and so this can create some disruption for the binding of the address system to the receptor. To synthesize pyridazinone modified in carbon 6, we can start from the methyl ester of 5-aminoleucinic acid, which is Boc protected, or from 4-ketopimelic acid. Both these reagents can be reacted with hydrazine to give the pyridazinones, which can then be translated via reaction with a carboxybenzaldehyde in position 4 in a condensation reaction, and alkylation in position 2 with a chloralkane. We obtain an address system that in 6 has a carboxylic acid or an amino function, which can be used to introduce fluorophores and so to develop optical probes. For example, the carboxylic acid has been reacted with an amino body P that we synthesize in lab to obtain this optical probe that was presented in the previous slide. The same functions, carboxylic and amino, can be used to introduce linkers and then the fluorophore to keep the fluorophore distant from the address system. We have then developed a new series of optical probes based on these results and using here fluorophores which are highly optically efficient. So these are all fluorophores that are bright and so enable optical microscopy studies. Also in this series we used linkers of various lengths to be sure that the fluorophore is at the right distance not to interfere with the binding of the address system. By measuring the optical properties of these probes, we found that generally in most of the cases we found high values of quantum yield and molar extinction coefficient. We have tested these compounds also for affinity for FPRs in our calcium flux assay and found that all the probes are highly active. The activity is in the nanomolar range, and two probes, B2 and B3, are the most potent with an activity of about 2 nanomolar on FPR1. Also, these compounds showed selectivity as agonists towards formylpeptide receptor 1, as they are not able to act as agonists on formylpeptide receptor 2, so are not active as agonists on formylpeptide receptor 2. So with this probe, we can now perform optical studies, and we can, for example, measure directly the binding and the affinity for FPRs using the fluorescence as a readout. This can be done in flow cytometry or can be done on a plate reader. And this represents a more direct way of measuring the binding, the affinity for the receptors compared to functional tests. Also, we have started some microscopy experiments with these probes by incubating formylpeptide receptor 1 transfected cells, so HL60 cells, with the probes. Here is reported the microscopy with the probe B2, which was incubated with the cells, and then the cells were fixed. As we can see from these microscopy images, we can detect the probe with the cells, so indicating that there is binding to the cells and potentially to formylpeptide receptors. And also most of the signal is in the cytoplasm. So we believe that the probe, by interacting with the receptors, determines the internalization in the cytoplasm of the receptors. This is additional microscopy with the other probe, the compound B3, which was incubated with the same FPR1 transfected cells. And in this experiment, the cells were imaged live. Also here we can detect the probe, and we can see that the localization is mainly in the cytoplasm, although it looks not evenly distributed. If we look at these enlarged images of these cells, we can see that there is low signal or no signal in the peripheral part of the cells. Most of the probe is perinuclear and has a punctuate nature. So this suggests endosomal trafficking, and so this is in favor of thinking that internalization of the receptor by binding with the probes, so the probes determine internalization of the receptors. To further confirm the specificity for formyl peptide receptor 1, we performed additional microscopy tests. Here is reported the test with a formyl peptide receptor established antagonists that we used, the compound BOC2. And what we see here is that when the cells are pre-treated with the antagonist and then we apply the probe, we lose the signal, the fluorescent signal coming from the probe, because the probe is unable to bind to FPR1 due to the presence of the antagonist. Differently to the case when we use only the probe and we can record fluorescent signal within the cells. So this confirms specificity for formyl peptide receptor 1, and also was this was also confirmed by flow cytometry studies, where we could see that for cells treated only with the probe, we can record a high signal, whereas cells pre-treated with the antagonist and then applying the probe, we have very low levels of fluorescent signal. For the internalization, we used some blockers of internalization. Here is reported the microscopy experiment with the compound DINGO4, which is a blocker of internalization. So with this compound, the internalization cannot happen. As we can see in this video, by pre-incubating the cells with DINGO4 and then applying the probe, we lose the signal in the cytoplasm. But we can detect signal on the cell membrane, where originally the formyl peptide receptors are located. So this proves that the internalization is blocked and we don't have signal in the cytoplasm, but we can record signal where originally the formyl peptide receptor are located, so on the cell membrane. Also, in this experiment using the probes, we were able to see, if you look at this image here, we were able to see changes of the shape and the size of the cells that were detected with the probe. Also, this change of shape and size is a typical effect of G protein couplet receptor stimulation with an agonist. So we have developed a library of probes which are specific for formyl peptide receptors, and we will continue to use these probes to determine additional cellular effects which are influenced by formyl peptide receptor activation. Also, we plan to perform in vivo studies using in vivo models of inflammation by labeling human neutrophils with these probes, and then we can track the movement and assess the function of these immune system cells in inflammatory events. In the second project that I'm presenting, I will discuss our results on the development of imaging agents for alpha V beta 6 integrin in the context of pancreatic cancer. So integrins are also membrane receptors which facilitate cell adhesion, motility, and survival. The isoform alpha V beta 6 is highly overexpressed and upregulated in solid tumors, mainly pancreatic cancer, whereas the expression is minimal in healthy cells. There is general consensus that alpha V beta 6 is involved in tumor progression and metastasis, and can be considered a biomarker for pancreatic cancer. And so is a relevant target for developing an imaging procedure. Just to mention that pancreatic cancer is a cancer with a very low survival rate. And this is mainly due to late-stage diagnosis and the incomplete removal of the tumor during surgery. So the development of optical probes that help visualizing the tumor mass in this type of cancer is highly beneficial. To target alpha V beta 6, we have selected this cyclic peptide, which was reported as a specific binder of the alpha V beta 6 integrin isoform, and as an activity in the subnanomolar level. This peptide contains the RGD motif, which is a typical binder these three amino acids, RGD is a typical binder of integrin receptors. The peptide was reported to have high selectivity for alpha V beta 6 isoform and high stability in vivo. Both the high selectivity and the high stability are due to unnatural modifications which are present in the peptide. So first of all, the RGD is included into a cyclic structure, and also there are unnatural amino acids, for example, a deep proline and a methylated lysine here, which prevent the hydrolysis from proteases. So we have used this as an address system to develop alpha V beta 6 targeted imaging probes. The synthesis of this cyclic peptide is performed in solid phase peptide synthesis, or we have also set up a new method at the peptide synthesizer to speed up the production of the peptide, and so obtaining the cyclic peptide in about 2 days. We use FMOC chemistry to obtain this peptide. So via coupling of FMOC amino acids consecutively, we can obtain this linear peptide on a chlorotriethyl resin. Then the peptide is cleaved from the resin using an acidic solution, and the linear cyclic peptide, the linear peptide can be cyclized by reacting the amino group on the aspartic acid with the carboxylic group on the glycine using diluted solution of the linear peptide. In this way, we develop the alpha V beta 6 address system, which has a lysine with a free amino group on the side chain that can be used to introduce an imaging tag. We have developed PET imaging agents specific for alpha V beta 6 in the context of pancreatic cancer, using this cyclic peptide as the address system. And so in the cyclic peptide, we have incorporated chelators for radiometals as the DOTA or the in-house produced THP. We use the THP because THP belongs to a family of chelators which are validated chelators for clinical use. The THP is based on the unit of 3,4-hydroxypyridinone, which are strong chelators for three basic metal cations. And for example, the gallium, that is the radionuclide that we want to use for PET imaging agents. So the hydroxypyridinones form hexadentate complex with three basic metal cations in a 3-to-1 complex. If the 3 units of hydroxypyridinone are included in a tripodal structure, as in the THP, the affinity for the metal is even higher. For the synthesis of the THP, we start from nitromethane, which is reacted in a Michael addition reaction with terbutyl acrylate to obtain this 3-branched nitro derivative. And then the nitro group here is reduced to amino, and the amino group is protected with a lock. After removing the terbutyl protections, the free acid can be coupled with the hydroxypyridinone to give a fully protected THP. By removing the benzyl protections on the THP and the alloc protection on the amino group, we obtain the free THP that chelates gallium with high affinity. Also, the amino group in this position can be used to introduce the linker and also to bioconjugate the address system. So we have developed these PET imaging agents. 2 are based on the THP and 2 are based on the DOTA, using in this case 2 different radiometals, so gallium-68 and lutetium-177. We have tested the efficiency of these chelators for chelating radionuclides, and so efficiency to be translated in PET imaging agents. And here are reported the radio TLC and the radio HPLC results showing that in all cases we find a single peak that indicates that we have a quantitative complexation when the ligand plus address system is combined with the radiometal used at each time. And also we find in all cases a high radiochemical purity, which is higher than 95%. So these compounds are effective as PET imaging agents. We have tested the affinity on alpha V beta 6 by using pancreatic cancer cells, so BXPC3 cells, which are pancreatic cancer cells overexpressing the alpha V beta 6 integrin. And we found that the DOTA analog has an activity in the low nanomolar range, so it's highly active. Whereas the THP analogs, they have an activity in the sub-micromolar range. Also, testing the specificity for pancreatic cancer cells, we have incubated one of our probes here in this plot. One of our probes was incubated with different cancer cell lines, so BXPC3, which are pancreatic cancer cells, HCC827, which are lung cancer cells, and CT26, which are colon cancer cells. We can see that the majority of the uptake happens in the pancreatic cancer cell lines, giving an idea of specificity for this type of cancer cells. Also, the DOTA analog, which was the most active, was tested in a clinical study in a hospital in Germany to visualize ex vivo via PET imaging tissues from pancreatic cancer patients. The compound resulted effective as a PET imaging agent ex vivo, but when tested in patients, we found a rapid renal excretion, and consequent to the rapid renal excretion, we found a minimal uptake into the pancreatic cancer sites. We think that this rapid renal excretion is mainly due to the high hydrophilic nature of this radiotracer, because the DOTA is hydrophilic and the peptide is hydrophilic. So the high hydrophilic nature limits the use in vivo of this PET imaging radiotracer. So we are now focusing mainly in performing more modifications on the THP series, first of all, to increase the affinity for the alpha V beta 6 integrin, and also because this series maybe can have a better profile in terms of biodistribution. So we should consider that in the THP we have already three residues of hydroxypyridinone, which are aromatic, and so they confer some lipophilic character to the molecule. Also, we can further increase the lipophilicity by introducing lipophilic functions in the linker in order to improve the biodistribution properties. If we are able to identify a PET imaging tracer based on THP and gallium-68, this can also be used to chelate gallium-67, and so can open possibilities of combined use in therapy and imaging. In the last project that I'm presenting, I will discuss the development of oligomeric complexes between iron and fatty acids for the treatment of anemia. So anemia is a disease in which the body has a low number of red blood cells, or has red blood cells that are abnormal. And the causes of anemia is the deficiency of some nutrients, for example, vitamin B12 or B9, or can be various temporary or chronic conditions. The most common form of anemia diffused worldwide is the iron deficiency anemia, that from the name is due to low levels of iron in the body. So low levels of iron give low level of hemoglobin, and in turn, low levels of hemoglobin give a reduction of the number of red blood cells, or red blood cells which are abnormal. The treatment of iron deficiency anemia consists basically in bringing up the levels of iron in the body. And this is done via taking some iron supplements. as, for example, this product reported in the slide. These preparations are composed by an iron 2 salt plus ascorbic acid. The iron 2 salt can be in the form of iron phosphate or iron sulfate, and the ascorbic acid is needed to keep the iron in the oxidation state too, because iron 2 is considered more absorbed and more bioavailable. The problem with this preparation is that they give a low compliance or no compliance by patients due to several side effects at the level of the gastric tract. So patients tend to leave the treatment quite early. So in collaboration with this company, Rina Pharma, we have decided to look at an alternative preparation for iron deficiency anemia. Here are reported some considerations in between the use of iron 3 and iron 2. So iron 2 is highly soluble, and this is the reason why iron 2 is used in all the current preparations, all the current supplements used to treat iron deficiency anemia. Differently, iron 3 is not soluble and forms insoluble products at pH higher than 3. So all the preparation attempted based on iron 3 failed due to poor bioavailability. The problem of iron 2 is that it is redox active, so iron 2 can rapidly oxidize to iron 3 at physiological pH, generating reactive oxygen species, so generating free radicals, for example, the hydroxy radical that is generated through the Fenton reactions. If we consider that each one of these preparations, so a tablet of these preparations contain about 60 milligrams of iron sulfate, of which only 2 milligrams of iron are absorbed, we can see how the amount of iron by taking one of these tablets, which is free and potentially can give oxidation, is very high. Differently, it's generally agreed that iron 3 does not produce free radicals, so it would be ideal to develop a preparation based on iron 3, but we need to make the iron 3 bioavailable. So we thought about making complexes between iron 3 and long-chain fatty acids, as these can help in the transport of iron across cell membrane. And this is because fatty acids are normally absorbed in the small intestine, which is the same place where iron is absorbed, and fatty acids are absorbed as emulsion or micelles. So potentially, if we have iron-loaded fatty acids, so fatty acids loaded with iron, the iron could be absorbed through the same emulsions and micelles, and so can enter the circulation. The other possibility is that the complex iron fatty acids breaks in contact with the intestinal barrier, and but at this level, once in the small intestine, there is still possibility for the iron 3 to be converted in iron 2 and then be absorbed. To develop iron 3 long-chain fatty acids, we considered that iron forms oligomeric complexes with carboxylic acid. This is a feature not only of iron, but also of several other 3 basic metal cations. As an example, here are reported the oligomeric complexes of iron 3 with citric acid and manganese 3 with acetic acid. Also, if we are making a complex iron fatty acids which has the tendency to form emulsion at physiological pH, we considered that long and saturated chain fatty acids should be preferred as the lauric acid and stearic acid that we have selected. So we have started some synthetic work to generate these oligomeric complexes, and we used in these reactions an iron salt, for example, sulfate, chloride, or nitrate, as sources of iron. And these were reacted in water or with an organic solvent were reacted with the fatty acid or the sodium or potassium salt of the fatty acids. We have obtained oligomeric complexes of this general structure here, where we can see that there is a 3 nuclear iron centers. So 3 irons are in the center of the complex bound together, and then each iron is bound to 6 oxygen ligands. The oxygen ligands come from the molecules of fatty acids, water, which is also coordinated to the iron, and a triply bridged oxygen in the middle of the complex. The 3 molecules of water coordinated to the irons can be substituted depending on the condition of the reactions and the solvent used by molecules of other solvents. For example, here we have ethanol in the place of water. Also, this complex has a positive charge, and so depending on the salt used to make the reaction, the counter ion can be nitrochlorine or the fatty acid itself. Here is reported the structure of one of these complexes, the iron trilaurate. To characterize these complexes, we mainly rely in elemental analysis because NMR is not possible, and mass spectrometry or IR would give partial indications. But in some cases, we have been able to obtain from ethanolic solutions of this material at -20 degrees crystals out of this structure. So we have crystallized this material, and the crystals are not stable at room temperature and have a melting point of -5 degrees. So here is reported the crystal structure that we have analyzed in the X-ray, where we can see that we confirmed the predicted oligomeric structure for the complex. We have 3 iron centers and a triply bridged oxygen in the middle, and then the irons are coordinated to the fatty acid residues and 2 molecules of ethanol in this case. 2 additional molecules of ethanol are present in the crystal of this material, forming an hydrogen bond with the nitro counterion. This is the unit cell for the complex to show that we have 2 molecules of iron laurate in each unit cells, and also to show that already in the solid and crystalline state, we can see there is tendency of this material of aggregating into particles due to the hydrophobic interaction of the aliphatic chains of the lauric acid. By performing some formulation studies to check if the complexes had tendency to form emulsions, we have mixed the iron laurate with the sodium laurate at different %. And we use sodium laurate because sodium laurate can contribute to the formation of emulsion and also can minimize the dissociation of the laurate ligands from the iron complex. So in a way can preserve the complex in biological environment. From the microscopy here, we found that we have an homogeneous size distribution for the complex with particles that have a diameter that for 90% of the particles is lower than 2.4 micrometers. We tested the complex first in cells, and we used Caco2 cells, which are a good model to mimic the intestinal cells. And in this experiment, we have loaded the cells with 2 formulations of iron laurate at 2 different % of sodium laurate. After incubating the cells for 4 hours with the complexes, we can see that we have an increase of the amount of iron inside the cells. So we are able to provide iron to the cells, and this increase of iron recorded in the cells is in line with the increase of iron that is determined by iron sulfate, which is our reference standard. Also, we have started some in vivo study recently using anemic CD1 mice. These are mice that are made anemic by keeping them on a low iron diet for several days. And then in this experiment, we wanted to check the capability of the iron fatty acid complexes to regenerate hemoglobin, so to replete hemoglobin in anemic mice. In this plot here is reported results obtained with the iron stearate complex. We can see that at the beginning of the experiment, all the mice are anemic, all the treated mice are anemic, because compared to untreated mice, there is a low level of hemoglobin in these mice. After 10 days treatment with iron stearate, we have an increase of hemoglobin, which is significant. And this increase of hemoglobin is similar to the increase of hemoglobin determined by the iron sulfate, our reference. A similar result was also obtained with the 2-iron laurate formulations. So in anemic mice, after 10 days treatment, we have an increase of the hemoglobin levels by treating the mice, supplying to the mice iron laurate at 2 different percent of sodium laurate. And this increase is pretty similar to the increase of the reference material iron sulfate. So we are able to provide iron to anemic mice using this formulation, iron laurate and iron stearate. We are now continuing the pharmaceutical development by focusing mainly on iron laurate, because iron laurate is an easier chemical to work with. Instead, iron stearate is insoluble in most common solvents, and also is difficult due to the fact that it's not soluble. It's difficult to be formulated or dispersed in to make a preparation. So we are mainly focusing on developing iron laurate at the moment, and with support from the company, we are about to start toxicity studies on this material to continue develop the product as a supplement for iron deficiency anemia. So I would like to say thank you to the people in the group that have contributed to these projects. And thank you to the collaborators for both the biology and the chemistry. And thank you very much to you for attending this seminar online. Thank you.
Dear Agostino, I am impressed with your work. You have done a lot of work in 3 distinct areas. I am very impressed. I am sure that all the colleagues are also very good, impressed with your work. And so I would like to ask to our colleagues if someone would like to make some questions to Professor Agostino. If no one wants to ask something to Professor Agostino, I would ask if, I think you are in the middle of your work, I think, yeah?
Yeah, most of these projects are all ongoing.
Yes, you have a lot of work, and I am sure that you have some very good students to help you with that work. Like was the case of my student, Renan.
Yeah.
And I think it's not possible to do work so deeply without good people to help you. You have the excellent ideas, but you have to put.
Yeah, this is fundamental. This is fundamental in research to have good people to rely on for making the projects happening. So what an idea is, and then to translate the idea in practice.
Yes, and I am sure that you get very good results in the future. You have already get good results with your work.
Thank you.
I'm sure that you are very persistent researcher, and with many good ideas and good students. I think that's normal in the King's College. Yeah, all the good students of the world go to King's College. And I would ask something particular. So you have some molecules that are very big, I think,
Mm.
big
Yeah.
molecules, but they are soluble in some solvent or water and so on. Yeah, they are
Yeah.
always soluble.
Well, you're referring, for example, to the PET imaging agents, for example, these molecules here. Can you see my slides?
No, not yet, not now.
Okay, for the PET imaging agents, this is the biggest molecules I have presented today. These are mainly based on. There is a message from Renan in the chat. So these are big molecules, but they are based on peptide structures. So they are yes, very soluble in water and in aqueous solutions.
Yeah, that's a very interesting point because uh, molecules so big molecules and they are also soluble. That's interesting.
Mm.
Yeah, that's interesting because I work with microreactors and we know a lot of
Yeah, I
microreactors.
remember we discussed this when we met last time. That to find a way of making via microreactor technology molecules of big size.
Yeah, it is possible.
Mm.
The problem is when we have not soluble substances, but if they are soluble, it's no
Yeah, these
problem
are
at all.
soluble based on peptide residues. Most of them are amino acids, so these are soluble in water solvents, aqueous
Yeah.
solvents.
I think Renan is there. I think Renan could make some good question for you. Not really, Renan? Could you ask something for your supervisor, ex-supervisor? Hi, Renan, are you there? I don't
Maybe
think.
he's not allowed to speak, but yeah, let me say again, thank you very much, Mauri, to you and to your department for organizing this seminar online.
It was a pleasure for us to have a very good scientist like you to give us a wonderful presentation with so many results in three different um, uh, directions. I can say you have a lot of work, and I am sure that you'll be successful. And if Renan would like to add something to Agostino, please go on, Renan. I don't know. Probably he can't make a question. So
When Renan was, it was a good job and very interesting. When Renan was working in my lab, he contributed to make these hydroxypyridinones that I have shown as the THP chelator. So the unit that chelates in THP is an hydroxypyridinone, and Renan worked at the synthesis of these hydroxypyridinones for chelating metals for clinical applications.
okay, I see the complexity of making a so big molecule and treat
Yeah,
different
Renan.
parts of the molecule and put one small brick in some point. I think is very complex matter to make chemically.
Renan
Yes.
is asking a question: can hydroxypyridinones can be used in different kind of cancer? Yes, Renan, this is a good question. So we have applied the use of hydroxypyridinone as chelators in various cancer. Indeed, in another project, we have a molecule that is based on hydroxypyridine for PET imaging to target prostate cancer, and this is also at clinical stage, so is in clinical trials at the moment for the PET imaging of prostate cancer, and is based on hydroxypyridinones as the chelators for radiometers for gallium-68.
Yeah, that's very interesting. So we can think in another possibility to Renan to go back to London and continue the work with you. It will be very good for us in the university, in the School of Pharmaceutical Sciences of the University of São Paulo, and also for Renan and for you, I'm sure.
Yeah. Also, I knew that Renan has just started a new job, so best of luck to him for this new job.
Okay, Agostino, I'd like to thank you very much for your presentation, and we hope that you have all your issues at home with your family, with your daughter, be resolved soon, and we would like to have you in Brazil, if possible,
Yes.
up to the end of this year. It is
Yeah.
possible. For our side, it's possible.
Yeah, thanks very much, Mauri, for this seminar and for the invitation. I will do my best to try to finalize this trip by the end of this year. We will be in contact, and we can select some dates where I can visit your group and department.
Okay, thank you so much.
Thanks very
much. Hope to see you soon.
And thank you everyone for attending.
Bye bye.
See you soon. Bye bye.
Bye bye.